Till now we have understand the tissue and tissue system of the plants. Now we'll understand about the anatomy of monocots and dicots with the help of examples. The various plant organs like root, stem and leaf show different tissue organization in their anatomy, which can be observed by transverse and longitudinal sections within their stain preparations. Let us understand each of them in detail with the help of examples. Let us first of all begin with the anatomy of dicot roots. This figure clearly depicts about the anatomy view of dicot roots like sunflower. It consists of thin-walled parenchymatous cells with intercellular spaces present. Then below cortex arises the endodermis, which is considered as the innermost region. It is similarly made up of parenchymatous cells but closely arranged without any intercellular spaces. Upleaf. All the tissues that are present beneath the endodermis, like pericycle, xylem, phloem, constitute this stain. Now let us understand the functions of each of the layer in brief. First of all, let us begin with the outermost layer, that is epidermis. Epidermis of root is also known as epiderma and it forms the outermost layer of the cell, which is made up of parenchymatous cells. From here, thin thread-like projections appears which are known as root hairs. These unicellular root hairs increase the absorptive surface and helps in the absorption of water and dissolved mineral. Then the second layer below epidermis is cortex. It is also made up of small parenchymatous cells with intercellular spaces present. Beneath cortex appears endodermis, which is considered as the innermost layer of the cell. These are also made up of parenchymatous cells with no intercellular spaces present. And so these barrel-shaped cells are arranged in a compact manner. These radial walls of cell shows impermeability to water and is arranged in the form of impermeable wax like suberin as well as lignin in the form of thin strips which is called Casparian strip. Below endodermis arise the fourth layer known as pericycle. These are a few layers of parenchymatous cells which are present beneath endodermis. From here, the lateral or secondary roots arises. Below this layer comes pith. Pith is considered as the centralmost or the core part of the cell. These are also parenchymatous cells which are present between xylem and phloem, which is known as conjunctive tissue. Then the region appears below pith is vascular bundle. These vascular bundles are actually a layer that is formed between endodermis and pith. Vascular bundles involve xylem and phloem. There are usually 2 to 4 xylem and phloem patches. Now, all the tissues inside endodermis such as pericycle, vascular bundle and pith is commonly together called as stem. Example, on studying the anatomical view of dicot root of sunflower, that is, observing the transverse section of sunflower root with the help of stains like safranin, the stain appears radial, alternate and tetrarch in nature. Till now we have studied about the anatomy of dicot root such as sunflower. Now we will study about the anatomy of monocot root like maize. This figure depicts the transverse section of monocot root like maize. As it can be seen from the figure, the outer layer is known as epidermis. which is made up of parenchymatous cells. From epidermis, thin projections develop which is known as root hairs. Like dicot root hairs helps in the absorption of water and mineral salts. Now below epidermis arises a layer which is made up of parenchymatous cells known as exodermis. When the epidermis layer gets destroyed, then this exodermis layer becomes functional, which is protective in nature. Below exodermis arises a layer known as hypodermis. This is made up of sclerenchymatous cells, which provides mechanical support to the plant. Then, similarly like dicot root, further epidermis is followed by cortex, then endodermis, pericycle, pith, con- in between which vascular bundles are located. Let us understand the function of each of the layers in brief. Thus, from the figure studied above, it is clear that many layers of monocot root is similar to that of dicot root. So, let us begin with the first layer, that is epidermis. We have already studied that epidermis is the outermost layer made up of parenchymatous cells. Further, following epidermis, there comes another specialized layer known as exodermis, which is only present in monocot roots. This exodermis becomes active only when epidermis loses its function. Then exodermis becomes active. This exodermis layer is also made up of parenchymatous cells. Below exodermis, there also arises another layer known as hypodermis. This layer is made up of sclerenchymatous cells which provide mechanical support to the plant. The other layers of monocot root that are similar to that of dicot root performs the same function as discussed earlier. However, as compared to dicot root, which possess 2 to 4 patches of xylem and phloem, here fewer xylem bundles are present, which are more usual than 6 arc xylem bundles. This means that when the protoxylem of xylem lies towards the peripheral side or outermost region near to the endodermis and metavaxylem inside lies towards the pith, then the condition is known as exarch, which is possible or which situation arises only in monocot roots. As compared to monocot roots, as compared to dicot roots, monocot roots possess pith, which is larger and well developed. Like dicot roots, stem present is also radial and alternate. However, unlike dicot roots, here the stem present is polyarch in nature, unlike dicots where it is tetrarch. This figure clearly depicts the exarch nature between the dicot and monocot. Here, the pith region consists of tetrarch stele. On the contrary, here it consists of polyarch stele.